So now we continue with the next chapter, the chordate development. Chordate development starts with fertilization all the way to post-embryonic developmental processes like aging and senescence. Development never stops. But this is the purview of developmental biology, the details of which. In 102.1, we only study the basic features of early embryonic development from cleavage all the way to formation of the neural tube or dorsal hollow nerve cord. So as defined, cleavage is basically just several rounds of cell division without much increase in cell size. Because of that, it produces numerous smaller daughter cells called blastomeres. Several cleavage types are seen in chordates depending primarily on the amount of egg yolk, which impedes the cytokinesis, yung cleavage furrowing process. So kapag konti yung yolk, microlecythal egg kagaya ng Amphioxus and sea urchin, wala masyado nag-i-impede ng cell division, therefore the cleavage is holoblastic, meaning whole, and the resulting daughter cells are roughly equal in size. Kapag may moderate amount of yolk kagaya ng amphibians, yung side na kung saan mas maraming yolk, mas malalaking cells kasi nai-impede yung cell division dun, whereas the side kung saan mas konti yung yolk, mas maliliit yung cells kasi mas mabilis yung cell division. So the cleavage type is holoblastic but unequal. Kapag sobra, the extreme case is macrolecythal, kagaya ng sa reptiles and birds. The cell division is incomplete or meroblastic. A special type of meroblastic cleavage is discoidal, wherein the embryo, the cleavage process, happens only within a small spot on top of a huge yolk. It's a small disc, the blastodisc, kaya discoidal. In the lab, we use the sea urchin embryo as a model for microlecythal egg development instead of the Amphioxus. The culmination of cleavage is a hollow ball of cells called the blastula. The cavity inside here is the blastocoele. The cells, products of cleavage, even as early as the two-cell stage. These are called blastomeres. This is the cleavage in specimen in frog embryo. Because of the presence of yolk, cell division is faster in the animal hemisphere, where there's relatively little yolk. Blastomeres here are smaller compared to the vegetal hemisphere, kung saan naiipon yung yolk. Mas malalaking blastomeres dyan. Again, a blastula is formed hollow ball of cells. The cavity is the blastocoele. The animal hemisphere marks the future dorsal side of the animal. Gastrulation is a time of major upheaval when cells start to move around and differentiate into the three primary germ layers. The beginning of gastrulation is usually the formation of a blastopore. Mag-i-invaginate yan. Nagkaroon ng pore, where the outer cells start to move inside, creating the primitive gut or the archenteron. Being a deuterostome, the blastopore eventually becomes the anus and marks the posterior side of the animal. So during gastrulation, the major body axes are established and the germ layers that will give rise to most of the organs are in place. The outer cells become the ectoderm. In sea urchin, some cells migrate into the blastocoele. Yan, yung mga small spots na yun. Those are the migratory mesenchyme cells, basically the equivalent of a mesoderm. And then the innermost cells composing the archenteron itself is the endoderm. This is just early gastrula. As you can see, nagpo-form pa lang yun, o nagsa-start pa lang yung invagination process. And then in the frog, gastrulation also starts by formation of a pore, the blastopore. The dorsal lip of the blastopore forms first. The ventral lip forms later, kaya mas fully formed lagi yung dorsal lip of the blastopore. The archenteron here displaces the blastocoele. Pag nag-invaginate niya, nabi-displace or napapalitan niya yung first na cavity, which is the blastocoele. eventually ma-obliterate yung blastocyst. Then the involuting cells from the dorsal blastopore lip and becomes the roof of the archenteron. This will become the chorda mesoderm. This is the chorda mesoderm. These cells here lining the roof of the archenteron. And then eventually the chorda mesoderm will give rise to the notochord, as you know. These involuting cells will also spread out laterally and then ventrally, to give rise to the rest of the mesoderm layer. So there's ectoderm, mesoderm, and then this huge chunk here is the endoderm. Initially, a yolk plug is wedged in between the blastopore lips, but will be resorbed as the embryo grows. So ma-absorb din siya as the embryo uses the nutrients of the yolk. In those with macrolecythal eggs, cleavage only happens within a relatively small spot, as I said, the blastodisc on top of the yolk. Kaya discoidal ang cleavage nila. The huge yolk also places constraints to gastrulation. Blastopore doesn't form. Instead, what they have is a primitive strake, which is formed as a thickening of the outer layer of cells. So magti-thicken yan, mag-start siya from the posterior margin of the blastodisc. And then hahaba siya ng haba anteriorly. Yan, magiging streak siya. And then eventually, mag-determinate siya into a Hensen's node or primitive node. These structures represent regions where the outer epiblast cells start to migrate inward to create the mesoderm and endoderm layers below. So instead of a blastopore, that's their way of performing gastrulation. Neurulation happens while gastrulation is still in process. There are 3 specific stages. So there's the neural plate, flattening pa lang ng dorsal ectoderm cells. And then nagfo-fold siya, also called as the neural groove stage. And then eventually magpo-form na siya ng tube, the neural tube stage, forming the dorsal hollow nerve cord. There's the neurocele. And then there's the neural tube, and then the notochord. You have the endoderm. The mesoderm is in between. And then the ectoderm. At this stage, neural tube stage, differentiated na din yung mesoderm mo. So from dorsal to ventral, you have the epimere, you have the mesomere, and then the hypomere. The hypomere splits into an outer somatic layer and then an inner splanchnic layer nakatabi ng endoderm. And then the cavity formed inside is the coelom. So I won't be discussing the germ layer derivatives in detail. You can read it on your own. Hyman provides plenty of examples, but you may consult other textbooks, of course. It would be enough to be able to enumerate 1 or 2 major organs derived from each of the germ layers.